Hey guys, Jayon King here. I'm here tonight with Chapter 4. I hope studying is going well for you guys. You guys still have some time left, so good luck with that. Yeah, so Chapter 4. So we opened Chapter 4 with talk of HM. He was a child patient with a severe case of epilepsy, and when doctors tried to treat this epilepsy, they removed his medial temporal lobe, parts of his medial temporal lobe. They removed the hippocampus and the parahippocampal region. When doctors did this, they found that HM wasn't able to develop new memories, but he still retained all of his past memories. When doctors told him new things after the procedure, he couldn't really remember them, right? But he still had a lot of memories from his childhood, whatever. So through this experiment, doctors were able to see that the hippocampus and the parahippocampal regions are important for converting short-term memory to long-term memory. So committing something to memory depends on those regions. So the easy thing to be confused about here is that people tend to think that the hippocampus is the area that stores memory, and that's not technically correct. The cortical areas break up your memory into different things and different components, and your memory is stored throughout your brain. The hippocampus and the parahippocampal regions are just for converting short-term to long-term memory. So, yeah, they might quiz that on the brain beast, so just be careful. So now, a talk of memory. So now we have to talk about the different types of memory now that we've actually talked about where memory is stored and how it goes from short-term to long-term. So our ability to consciously remember things, remember facts and events, is called declarative memory. And declarative memory has several forms. It has semantic memories, right? It has episodic memories, right? And you guys read about this. Semantic memory is just general facts and data. There are 125 million photoreceptors in each eye. That would be an example of semantic memory. Episodic memory, like you remember the time you first, I don't know, like walked. Okay, actually, well, that's pretty far back, but you get the point. So episodic memory, you remember a specific event in your life. Semantic memory, you remember general facts and data. And in order to use this information properly, you would have to have working memory. That depends on the prefrontal cortex. You bring up memories to your prefrontal cortex to use them, and that's working memory. So the ability to use declarative memory. So declarative memory is transient form, so to say. So as I discussed before, the medial temporal lobe is important for converting these memories into a long-term form that you can store and use. The other type of memory that you have to know is non-declarative memory. Non-declarative memory is procedural memory, so essentially how to do something. Most of the time, the cerebellum has a big part to do with this because a lot of the procedures that you do are motor things. For example, when you remember how to make turns on your car, for example, that would be an example of non-declarative memory or procedural memory. And your cerebellum would be really important to processing the timing of your movements. The basal ganglia also has a certain role to play with this, and this makes sense because the basal ganglia is where dopamine is produced, and dopamine is important for movement. Now, so we've talked about memory on a larger scale, but now we have to look at memory on a really, really small scale, maybe at the cellular or the molecular level, right? So when scientists first began to look at memory on a cellular or molecular level, when they began to look at this closer, they used the sea slug for research, Aplysia californica. And they managed to find the process of LTP, or long-term potentiation. So basically LTP, or long-term potentiation, is just a series of molecular interactions that essentially makes the connections between your neurons stronger. So that's how your memory is improved. So LTP begins when calcium ions are released into the synapse, and this triggers CAMP, or cyclic adenosine monophosphate. You guys probably learned about this in AP Bio. CAMP will then trigger another molecule called CREB, or CAMP response L-binding protein. It triggers a release of CREB, and then CREB will then trigger the release of neurotrophins, and the neurotrophins have the effect of making the synapses grow and thus making the connection stronger. You guys learned since day one that synapses are pretty much like the basis of your brain's connections. It's how your brain functions at a cellular level. So you can understand how important this is. So again, calcium ions are released into the synapse. This triggers the release. This triggers cyclic AMP. Cyclic AMP will trigger Krebs, and then Krebs triggers neurotrophins. And this happens at the NMDA receptors. And as you guys learned in Chapter 1, NMDA receptors, or N-methyl D-aspartate receptors, are where glutamate work. And so NMDA receptors are crucial to memory. So that's the talk of memory. Now, for language. Language. So language is really important to us. Obviously, we have to communicate, right? We discussed about like hearing people speak in Chapter 3. Language is really important even right now. I mean, I'm speaking to you guys. But language doesn't. Sometimes the language functions fail, and that's when you get a condition called aphasia. Aphasia is essentially a language disorder. So the book mentioned 3 different types: Broca's, Wernicke's, and word deafness. So Broca's aphasia happens when you get damage to the left frontal lobe, and as you guys learned in Chapter 1, the frontal lobe has the motor cortex, which controls your movements, right? When you speak, your mouth moves, and that's part of motor coordination. So when you get damage to the left frontal lobe in Broca's aphasia, your ability to speak is impaired. So that's the critical thing to remember about Broca's area. Speaking is impaired. Wernicke's area. So Wernicke's area is when you get damage to the left temporal lobe. As you guys learned in Chapter 3, the temporal lobe has a role in processing your auditory inputs. So when you get damage to the left temporal lobe in Wernicke's aphasia, you have the main function that's inhibited is your ability to understand what people are saying. So these are the main parts of those 2 aphasias. Now, of course, there are other consequences. For example, in Broca's aphasia, it has been shown that people have difficulty understanding more structurally complex sentences, and in Wernicke's aphasia, it has been shown that people tend to have errors when they speak. They tend to stutter or use more, or they tend to stutter or they tend to use more unintelligible gibberish words. But the main consequences are that in Broca's aphasia, your ability to speak is impaired. In Wernicke's aphasia, your ability to understand what people are saying is impaired. And when you get damage to the superior temporal lobes on both sides, then you have word deafness. So you can't comprehend speech on any level, pretty much. So Wernicke's aphasia people can still comprehend bits of speech, but when you have word deafness, you just can't comprehend anything at all. So as you can see, many different parts of the brain are important for the whole process of language in general. So the last thing I want to leave you guys with in this lecture is a talk of FOXP2. And FOXP2 is just the gene that gives us the ability to speak. And it's been shown that when this FOXP2 gene is mutated, people have a difficult time making the mouth and the jaw movements that are critical for speaking. So just know what FOXP2 is for the contest. So that's Chapter 4. Yeah, I'll be back tomorrow with Chapter 5. Hopefully, I'll be able to produce a video a day so I can get you guys all the information you need before the contest. See you around, guys.